So from the negative sense RNA viruses, we will study the Bunia virus first. The Bunia virus, actually, before we start the study of these viruses, let me tell you that we will study in three different portions. Every one we will study the first of all the general properties of the viruses, what kind of the properties, the general characteristics which are included with the viruses, then its replication, then its genome, that what kind of the genome it prevents, and what is the functions of the different proteins happening inside these viruses. So from taking the Bunia virus, it's the characteristics, the general properties. These are, of course, RNA viruses, enveloped RNA viruses. Actually, these found in the arthropods because these viruses need the vectors for its transmission. Vectors are some special kind of the animals or the organisms, or especially the flies and the rodents, which are needed for some of the viruses to transmit it to reach until the host. So the Bunia viruses are found in the arthropods and the rodents, occasionally infecting the humans. Sometimes they are also infecting the humans, but most importantly they are in the rodents and the arthropods. However, some of them are also infecting the disease in the plants. Vector-borne except the hantaviruses. As we already studied that these are the vector-bornes. Vector-bornes are those diseases which need some kind of the vector. Vectors are the intermediate host which are needed to reach the virus. These are not like the viruses we already studied, which just will go and will attack the host, and the host receptors will help them. These are the viruses which need the intermediate host under which the virus can replicate also, or maybe they are the reservoir for the viruses. So all of them, the Bunia viruses, are the vector-borne except the hantaviruses, which don't need any intermediate host. The vectors which are used for such kind of Bunia viruses, most of them are the mosquitoes or the flies, the sand flies using, the ticks, the special species from the tick-borne diseases. It's also important from the vector-borne diseases that they are the specific as far as their intermediate hosts are concerned. So in this case, the mosquitoes, ticks and sand flies can be used as the vectors. Now, why we already said that these are except the hantaviruses. What the hantaviruses will do for its replication? Hantaviruses are transmitted through the contact with the deer mice feces. The vector activity is very important as far as the transmission of the Bunia viruses are concerned. The vector, if there is some area where the most of the vectors are already there, then the Bunia viruses will be rapidly spreading and rapidly replicating. If there is some area, because it's not just like in the summer, there will be the more provision of the mosquitoes are there, so the Bunia virus will be more quickly transmitted. But if the winter there are no more mosquitoes, and its transmission will also be reduced. From the morphological point of view, they are spherical. 90 to 100 nanometer is their length. They are already, we have studied that they are enveloped single-stranded RNA. So they are lipid enveloped. The spikes, the envelopes also accompanied with the Skypes. The Skypes are there, and they are like matrix. They are metric proteins are there actually inside these. The are present are the large protein, N protein, and GC glycoproteins, and then GN proteins are there. And the lipid envelope is there, as it's clear from the diagram on the left side of the screen. Now, the classification of the Bunia viruses. Actually, as far as the classification is concerned, these are the largest family of the viruses, we can say. They have the 5 really very big genera which are causing the disease. And these are, as we already said, these are the vector-borne diseases. So vectors are very important. The intermediate hosts are very important for any purpose. Just like we can take the example of the dengue virus. We will study the dengue virus later. The dengue virus is transmitted through the mosquitoes. So if just the biologically point of view, we can just control the mosquito, we can just control the transmission, we can just control the transportation of the intermediate host. It will be very easy that we can control the disease. Similarly, so we are talking about the 5 of the genera of the Bunia viruses. Arthobunia virus, hantavirus, which we already studied that hantaviruses don't need the intermediate host, then the norovirus, fleovirus, and then tospoviruses. The diseases which are caused and how the diseases are caused by some of the special kind of the Bunia viruses. So we study them as far as the families are concerned. First of all is the family Peribunia viridae. It is causing the disease of the California encephalitis. And the species is, of course, the California encephalitis virus. Also, one more species is La Cruz encephalitis virus, Jamestown canyon virus, and snowshoe hare virus. So these are actually the encephalitis viruses. These are causing the encephalitis inside the host. And the vectors which are more important from the family Paribuniaviridae, and these are the mosquitoes which are acting as the intermediate host and which act as the reservoir of the viruses. Then the family Nairoviridae. Very important disease caused by the Nairoviridae, Crimean hemorrhagic Congo fever. And it is some part of the Africa which is very famous for such kind of disease. It is the neural disease. And the vectors are the ticks in this case and not the mosquitoes. This disease causes the fever, headache, eye pain, and the back pain. Also, maybe the itches are also there, and it is capable of just like the rabies, maybe rupture the backbone too. Then the diseases of the Pheniviridae. It is causing very important disease, the Rift Valley virus, the Rift Valley fever, and the vectors are the mosquitoes. Now the Hentaviridae. Hentaviridae we already studied, they don't need the intermediate host. So they will just use the feces of the deer feces, and the virus can be transmitted from one to other. The species or the disease caused by it are the hantavirus. The vectors are the aerosol excreta from these mammals. Actually, these are not the animal vectors. This is just the air, the aerosol excreta, which is excreted by the deer or which is excreted from the other mammals, and then it will be transmitted from the one to the other directly.